All right, so I will try my best to address your questions. In terms of the listeria, I actually had to pour a bunch of almond milk down the drain as well, and it was really upsetting because it's not cheap. But yes, that did come through this system that I'm going to talk about at the end of the presentation, are sort of emerging issues sort of process at the government. So I'm here. I'm Melanie Cousins. My married name is Gibson, but at work, since I'm Dr. Cousins, I stick with Dr. Cousins at work. And I'm going to talk to you about my life as an epidemiologist. So I currently work at the Public Health Agency of Canada in the Risk Assessment Division, where we conduct rapid risk assessments of emerging public health threats. But I'm going to give a bit of a background about myself before we get into the main topic. So I grew up on a farm outside of Burlington with my parents and two brothers. I now have an awesome niece and nephew, and I currently live in Guelph with my husband, Alistair, and our cat, RuPaul, and our dog, Waylon. Yeah, he lives up to the name, I will say. So I love to garden, read, camp, and hike in my spare time, and we also make our own hot sauce, jams, and pickles. I also have a side business where I make fun cupcakes and wedding cakes, but this has definitely taken a backseat now that I've graduated from grad school and I'm working full time. So this is the path that I took to become an epidemiologist for the federal government. And this is my PhD graduation at the University of Waterloo. So I started my journey with a Bachelor's of Science in Biomedical Science at the University of Guelph. And then after a brief interlude of traveling and working at Starbucks and finding myself, I started a Master's of Science in Epidemiology with an amazing professor, Dr. Amy Greer, who you may have seen on the news during COVID. She is one of Canada's top disease modelers, and she did a lot of the early COVID modeling. So during my master's with her, this was pre-COVID, we were looking at the environmental drivers of Campylobacter, which is a foodborne bacteria. And so I also built a transmission model of Campylobacter transmission in Ontario with flies as a mechanical vector, and how climate change could then change the impacts of these flies and the transmission of Campylobacter. I then started my PhD at the University of Waterloo with another amazing professor, Dr. Shannon Majewicz, and she's actually one of Canada's top foodborne epidemiologists and does a lot of consulting on foodborne illnesses in Canada. So in that project, we, along with a large international team, did work looking on the drivers of antimicrobial resistance from a systems lens. Overall, we were looking at the socio-ecological drivers of resistance and how we could address antimicrobial resistance under climate change. So then, during my time at Waterloo, I started as a student part-time with the government as a student epidemiologist. And I started in the foodborne and antimicrobial resistance surveillance division. So again, I was looking at antimicrobial resistance factors from the farm to fork continuum. I could do a whole other presentation on antimicrobial resistance, but I'm going to talk today mostly about what I do now. So when I graduated in 2022, I went full-time with the government, and then just about a year ago, I started with my current division, which is the Risk Assessment Division. That's a bit about me. But what I'm here mainly to talk to you about is what exactly epidemiology is, what do epidemiologists do, and what it's like working at my current role as an epidemiologist in the Risk Assessment Division. So a little bit about how emerging threats are assessed in the government. I also want to say that although I work for the government, everything I say today is my own views and understanding of the field of epidemiology and the processes at the government. I am still relatively new in the field. I'm only 2 years graduated from my PhD, so I'm continually learning on this topic as well. And I also want to mention that you can stop me at any time to ask any questions throughout, but there'll also be a question period at the end. So who has heard of epidemiology or epidemiologists? Great. And who had heard of epidemiology before the pandemic? Good. When I tell people I was an epidemiologist after my master's, pre-COVID, Nobody knew what I was talking about. Nobody knew what we did. So as devastating as COVID was, it was great PR for my field and awareness of what epidemiology is. And it's a really fascinating field. So in short, epidemiology is the study of the determinants, occurrence, and distribution of health and disease in a defined population. So if we break that down a bit, the occurrence of a disease is the trends of a disease. So how fast is it spreading? What is the current and historical patterns of disease? How is it changing over time? So here is a picture of the COVID cases in 2021 in Canada and the USA. So looking at the occurrence of the disease, it can help us understand our current situation compared to historical trends and also compared to other contexts. The determinants of a disease are the factors that may increase or decrease someone's risk of a disease. So for example, protective factors of COVID could be healthy diet and nutrition and the COVID vaccination. And then some risk factors could be age, different comorbidities, sex, and race. And again, this figure's from the internet. I'm not a COVID expert, but those are some examples of determinants or factors that can increase or decrease your risk. And finally, the distribution of the disease. So we can look at the distribution of a disease in terms of geography. So are there certain areas experiencing higher rates of disease? Is it more prevalent in urban settings? Is it more prevalent in dry or humid settings or areas of poor infrastructure? We can also look at the distribution among the population. So are there different rates of disease in people of different ages, different sex, socioeconomic backgrounds? So it helps us give a better picture of what is happening. So here's a distribution of COVID in Canada in May 2023 across different age groups. And so in 2023, we noticed that the highest rates are in ages 20 to 50. And this is people of working age. So that might be an area that we need to explore. And then we could maybe look at options for targeting communication. Yeah. Yeah.
When you say that the percentage by age group, is that percent of the population or percent of the age group?
Of the population. Yeah. Yeah, so we are seeing that like 20 to 29-year-olds are making up the most cases in 2023. And that could be because they're out working and interacting with people a lot more. They also might be the least vaccinated. I am not sure of that data specifically off the top of my head. I did look at it last year. But there's many reasons that, so these kind of things we see and then we go, okay, why is that happening? And then we look deeper into the data. So it gives us an idea of where to target for further exploration. So a little bit of trivia I thought I'd ask. How many of the top 13 causes of death in Canada do you think are due to infectious disease? Do you think it's the majority, like 10, or more like half, 6? So
Majority.
half majority. Okay, so it's actually only 3 out of the top 13. So COVID, influenza, and pneumonia are on the top causes of death. So we are actually very lucky in Canada in terms of infectious disease, both due to our climate. We're more north, we have a more cool climate, and a lot of diseases like more tropical climates, and also due to our good health infrastructure and sanitation. However, chronic diseases such as cancer, heart disease, diabetes, and Alzheimer's are the leading causes of death in Canada. And this could be due to many other factors such as our lifestyles, our sedentary lifestyles, or maybe our nutrition, our reliance on fast food, things like that. But I'm not a chronic illness expert, but those are just some hypotheses. But I do know that we are in pretty good standing in the world in terms of infectious disease. And so the last sort of main definition I thought might be of interest and causes a lot of confusion is the difference between endemic, epidemic, and pandemic. So a disease that is endemic means that it's regularly present in that area at a certain expected rate. So in Canada, we expect a certain number of cases of influenza each year. Or in Nigeria or many other countries in the Southern Hemisphere, malaria is endemic. And so we expect that it's going to be there. So when we travel, we take prophylactic treatment so we don't get sick. So that's endemic. An epidemic is a widespread occurrence above the average or above the expected amount. So we have epidemics or outbreaks of tuberculosis up north in Nunavut in Canada. So that's above the expected that we would see in Canada normally. There was the Zika epidemic that was in Brazil, or more recently, the dengue. There was a dengue outbreak or a rise in dengue in Brazil. And then there's pandemic. So a pandemic is when an epidemic then crosses boundaries. So it crosses the country boundaries and it spreads to multiple different countries. So again, we're very familiar with the COVID pandemic, and there was many other pandemics before it, including SARS, Ebola, HIV/AIDS. And I thought I'd put a little figure here. Loads. Oh. So I guess it's kind of small, but there's been multiple pandemics over the years that range in sizes. Some of the largest include the plague, or it was called the Black Death back in the 1300s, which wiped out 50 to 60% of the Europe population, which they estimated was around 50 million people. The Spanish flu in 1918 to 1920, that had a death toll estimated around 50 to 100 million people. HIV/AIDS started in the 1980s, but is still prevalent globally and is endemic in some regions, unfortunately. It is estimated to be around 33 million as of 2022, and then COVID has now made this top list with around 30 million estimated as of 2023. And then here's some pictures of Canada during some major pandemics and epidemics over the years. So there's polio, smallpox, and the Spanish flu. What these pictures are from. So since we're talking about history, I thought I'd mention John Snow. So John Snow is what we call the father of epidemiology. He was a physician in the 1800s who conducted the first recorded study that would be considered modern day epidemiology on record. He's probably not the first. So between 1817 and 1923, there was 6 large cholera epidemics. This outbreak was during the third wave of these. So in 1854, there was a large outbreak of cholera in London in the Soho region, and it killed 614 people in just a matter of a couple months. And since it was the 1800s, this was very early in the knowledge of pathogens and what pathogens were. But Dr. Snow hypothesized that what was killing people was coming from the water. So he then plotted the cases on a map, and this is the actual picture of the map that he plotted the cases. And he noticed that there was clustering of the cases. All the cases were happening in a certain region in London. So he decided to go door to door and ask a bunch of questions to these people or their families. And he found out that every single one of these cases had all been using the same water pump to get their water. And this is called the Broad Street pump. And you can still go visit it today because it's kind of an icon in our epidemiology world. So they did some digging, and they found that sewage was contaminating the water upstream from this pump. So they got the pump handle removed. People got their water elsewhere. Cases stopped happening. Outbreak was maintained. So this is considered the first kind of outbreak investigation. And epidemiology was used because he noticed an unequal distribution of cases. And then he used qualitative methods or interviews to pinpoint the common source or the risk factor or the determinant, what was causing the disease. So we owe a lot of our history to John Snow. So epidemiology can help us answer a whole bunch of different questions. I'm going to orient you to the figure here. So this is what we call the epidemiological triad. So in the middle, we have a disease. But in order for a disease to occur, we need to have an agent. So it could be a pathogen, such as a bacteria, fungi, or a virus. It needs to have the right host in order to survive. And that could be a certain type of person or animal or plant. And it needs to have the right environment to live and spread between its hosts. So using this triangle as a basis, we can answer a lot of different questions. So we can look at what is the cause of the disease? What is the source? What is the agent? Who is mainly being affected? Is it zoonotic? Is it transmission between humans and animals? What are the hosts? What are their risk factors or protective factors? And then we can look at how does it spread? What are the modes of transmission? Is it airborne? Is it foodborne, waterborne? How fast is it transmitting, and how far geographically is it transmitting? And then also what other factors are impacting the disease. So for example, is it impacted by different temperature or moisture or different climatic factors? And there's a lot of different methods we can use to answer these questions, including quantitative methods, qualitative methods, or mixed methods, which is a combination of quantitative and qualitative. So quantitative methods include descriptive statistics. So as we saw before, different percentages in the population, the distribution between different types of populations. So for example, it impacts 75% of the population or something like that. There's statistical models. So we regularly use, they're called linear or logistic regression models, and they can help us know the association between a factor and an outcome. So how much certain factors can change a person's risk or odds of disease. So this figure on the right here shows the results of a logistic regression, and it calculates the odds of death from COVID in different ethnic groups. So the odds of disease, this one in the top in the dark blue is adjusted for age. So that just means accounting for age differences. So if we adjust for age, the odds of dying of COVID are 3.5 times more in people from Bangladesh and Pakistan versus their white counterparts in England. This study was done. However, if we then account for differences in socioeconomic status, their regions that they live, household composition, so the structural integrity of their house and air quality and all that. If we try to adjust for all of those factors, they for some reason are still 1.75 times at odds of dying from COVID. So that warrants further explanation to figure out what is happening in these different ethnic groups that aren't due to differing age structures and differing socioeconomic backgrounds. So these kind of things sometimes answer questions and sometimes generate more questions. So we've accounted for what we thought it was. It's still more prevalent. So what is causing it? And then that would further more research. So then there's also other types of quantitative models. So there's transmission and projection models. So these simulate the spread of disease within a population and can estimate the number of cases that might occur under different scenarios. So I'm sure you heard a lot about these during COVID. They became very popular in the media to describe what is happening and what might happen. And then there's also qualitative methods, interviews, surveys, and these methods are great for getting in-depth information on why something might be occurring. So math can tell us what's happening, but interviews and more qualitative methods, we can talk to people and figure out why it might be happening and getting down to the why to help us then be able to change it. And then I thought I'd give a little bit more of a background on transmission models. I did do some work in transmission models and I thought they may be of interest. So transmission models, as I said, simulate a disease spreading through a population. The simplest are called compartmental models. So they split the population into compartments based on their disease state. So in this model, someone can be susceptible, exposed, but not yet infectious to others, infectious, or recovered. And sometimes it's called removed. So a susceptible person becomes exposed by coming into contact with an infectious person, and they have some certain rate of contact and some certain probability that that contact is going to make the susceptible person infectious or infected, sorry. Then after some period of time, they become infectious to others. Then after a certain period of time, you become recovered. And then if there's waning immunity, such as which happened a lot in COVID, the immunity doesn't last forever. You then can go back to the susceptible population. Yep.
Your model shows a dotted line from recovered to susceptible.
Yeah.
And I believe that's an epsilon.
Yeah.
And epsilon is used by mathematicians for a very, very small quantity. So what you're saying is those people who have recovered from the disease almost never catch it again.
No, I would say that is just a placeholder. It could be X, it could be Y, it could be any,
Okay,
yeah.
sorry, yeah.
Yeah, so there's usually some rate that we know after, say, 2 years, someone isn't like, or in COVID, because there were so many different variants coming around, it wasn't that like maybe a month and then you were infectious again, or sorry, susceptible again, and that rate can change if you're immunized. There may be a slower waning immunity, or it can last forever. And so if it lasts forever, we call that compartment removed from the population because they are no longer susceptible to getting it. And then they are removed from the population. Yeah.
Worried about the recovered people transmitting it to somebody else.
Yeah. Yeah. We try to account for that usually.
That's a fairly low.
Depends on the, it depends on the pathogen. So, polio, for example, you can be vaccinated and you can be immune from getting any symptoms, but you can still shed the virus. So if someone comes into contact with a virus that you've shedded, they can then become infected. So it's really pathogen dependent on how much recovered people can then go back to being susceptible and then influence and be infectious also. So these models can get crazy, actually. So that was just the most simple one. They're not often that simple, but that's usually a place that we like to start because sometimes we don't know what's going on. So they can account for many different things. They can account for population dynamics. So in the middle, that's an age structured one. So each age bracket has a different path through, and then they can mix with each other at a different rate. You can also account for different interventions like vaccination or quarantine. Here we have people being hospitalized. You can also account for pathogen dynamics. So in the top left one, we have the actual pathogen decaying in the environment. So then if you come into contact with it in the environment, after a certain amount of time, it may have disappeared, or you could use you could put in disinfectants into that model and say, oh, every 2 hours, someone comes around and disinfects, and that will kill the pathogen. And then you can also account for vectors. So in the top right here, we have a vector model. So like mosquitoes picking up the virus from someone and transmitting it to someone. So these models can get very, very complicated. But the thing you need to think about is that we need to have the data to be able to put into all these compartments and all these rates between the different compartments. So that's why we usually start simple and then we build out if we have data. So early in COVID, obviously we didn't have much data to go on. So what we tend to do with these models is we put in all the parameters or the rates that we know, and those are usually from the literature, from past studies. There's been studies on people watching how many contacts different people have. There's different rates for different age groups. People have done these studies. And then we try to, with all the data that we know, we try to run the model and see if we make the same, this is called an epi curve. So the same pattern of the data, and that validates our model. So if we make a model and then we run it on like a historical outbreak and it matches, that means we've captured the dynamics adequately. In early COVID, what they were doing is, I wasn't part of these models, but what I am assuming they were doing was using whatever rates they could at some sort of range and then fitting it to the data. So using the beginning of the curve to then go backwards and find the parameters. And so once it fit the curve, they could then project forward and see. But as more and more data came, you could see that the models were getting more and more accurate, or more and more better capturing the dynamics of what was happening. And then again, so then you can add different interventions. You can put like, what if we put in the masking mandate, and then you can see what would happen, or what if we, and then you try to model different things instead of having to do experiments on the population. So very brief overview of transmission models. Oh, sorry. And then there's even more complicated models. So there's agent-based models. So that's every person has their own characteristics. So you can have someone That accounts for, you can have someone start as a child and then they can age throughout the model. Each person can have their own behaviors. So people can go to work, people can go to school. But again, you need tons of data to be able to make those models. And then there's network models. So network models is everyone's contacts. So you can trace everyone's contacts and then you can put a disease in and see how it would spread through the network. And these are typically done for sexually transmitted infections because the contacts are more easy to account for than like respiratory, where you don't know who around you you're breathing on. And so but these models are very complicated and they need a lot of data. So they're usually done later, not in the emerging part, but later to understand what's happening. So you guys were all very in the know of what an epidemiologist was, but I would say pre-pandemic, when I said I was an epidemiologist, I would get things like, oh, so you work on skin, because like epidermis, epidemiology, it kind of makes sense. Or the people who had seen the movie Contagion thought that all epidemiologists were on the ground in an outbreak, dealing face to face with the infected and risking their lives every day and were superheroes. But that's only one type of epidemiologist. Those epidemiologists do exist. I am not one of those. I am a behind my computer type of epidemiologist at home in my office. So epidemiology is a very vast field. So those contagion type epidemiologists, those are the field epidemiologists. They're out in the field. They're talking to people. They're like Jon Snow. They're going to door. They're doing the interviews, and they're in disaster response usually. I am probably mostly an infectious disease epidemiologist, looking at how infectious diseases spread throughout the population. I was mainly a foodborne infectious disease epidemiologist. There's molecular epidemiologists. They look at the pathogen level. Are they transmitting genes between each other? How are they spreading? Very, very vast field. And so there's lots of ways that you can take what you've learned and apply it to other cases. Some of them you need other degrees, like a veterinary epidemiologist, you are trained vets, and medical epidemiologists are trained physicians first, and then they go into epidemiology.